So hello and welcome everyone to today's ALS United Greater New York's webinar on genetics and ALS. My name is Michelle McKenzie. I'm a care services coordinator for the New York City area. Thank you so much for joining us today. As you can see, today's webinar style, so we cannot see you or hear you. If you have a question, please put it in the Q&A box at the bottom, and at the end of the presentation, we will look at the box and answer those questions. So I have the pleasure today to introduce our presenter, Elizabeth Harrington. Elizabeth is a genetic counselor in the Eleanor and Lou Gehrig ALS Clinic at Columbia University Medical Center. She helped develop and now directs the ALS Families Project, which is designed to understand the genetic underpinning of familial ALS and the impact on affected families. So Ellie, thank you so much for joining us today. And with that, I'm going to turn my camera off and hand it off to you.
Great. Thank you, Michelle. Thank you, everyone who joined. As Michelle mentioned, I am a genetic counselor at the Columbia Medical Center ALS Center. And I have a presentation here today, a number of slides. Hopefully it will take up just about half the time that we have today, leaving us with a good amount of time for questions, and just a discussion around genetics. Genetics and genetic testing really should be a larger discussion with people with ALS and their families. So hopefully you guys, as the presentation goes along, just write in some questions and we can have a larger discussion at the end of our time. So I'm going to jump in here. Let me share my screen. Okay. Hopefully everyone can see that. If not, someone can jump in and tell me. What I'll be talking about today is really kind of a background on ALS genetics. So this might be a review for many people. We'll talk about very briefly how genetic testing is performed. Many of the people on the presentation today might be people with ALS or have family members who have ALS. So this is a talk really catered to you and this population. So what is your genetic risk or your family's genetic risk? And then considerations for genetic testing. So a lot of what I talk about today will really be what I cover in a genetic counseling session if you were to see me in a clinical session. So if you've had genetic testing with me, apologies for the redundancy, but perhaps there's some follow-up questions that will come about. All right. So quick genetics review, very basic overview of genetics so that we all have the same background here. And I know the last time many of us thought about genetics was in high school with the Punnett square, which I promise I won't get into. But in our bodies contains over, trillions of cells. Our cells contain our complete genetic code. So people will hear whole genome or whole exome. That just means our entire genetic code. And when we zoom in on our genetic code, that's kind of packaged or held on our chromosomes. You can think of those kind of like books that contain our genetic code. And when we unwind and zoom into our chromosomes, we can see the exact spelling of our genetic sequence or our DNA sequence made up of DNA nucleotides, A's, T's, G's, and C's. And certain stretches of our DNA sequence spell out specific genes. Genes are like specific sentences that spell out specific instructions or proteins that make our cells function and work properly in the body. So we have over 20,000 genes that tell our body how to function, how to work, give us specific instructions, hair color, eye color, protecting us against cancer. When we're talking about genetic testing for ALS, we're talking about testing specific genes. And you can think about genetic testing like reading through those genes with a spell checker, looking for a spelling mistake or what we call genetic variant or genetic mutation. And when we have a mutation in a gene, it means that gene doesn't work properly. the improper protein is produced, and we have dysfunction of the typical cell functioning, which leads to motor neuron degeneration when we're talking about ALS genetics. Okay, here's that Punnett square. But ALS genetics, the majority of our genes are inherited. All of our genes are inherited. The majority of the ALS mutations are inherited in what we call an autosomal dominant manner. So here on this figure, there's an individual with 2 chromosomes. Those are kind of the gray long rectangles. And in blue, we have 2 copies of all of our genes, one that we inherit from our mother, one that we inherit from our father. So for all of our 20,000 genes, we have 2 copies. And when we inherit our genes, here you have a partner. You either inherit 1 copy or another copy, and your partner passes down 1 copy or another copy. So our children, let me get my pointer, please. Our children are a combination of ourselves and our partner, and they only inherit 1 of our 2 gene copies. Okay, so most of these are dominant inheritance. If we have a mutated gene copy, our partner still passes down a normal or a normal gene copy, and we have a 50% chance of passing down a mutated copy or an equal 50% chance of passing down a normal working gene copy. Okay, so that's the majority of the ALS-associated genes. It's not all of them, but that's kind of what I'll be focusing on today. There's over 30 genes that have been associated with ALS. So here's our chromosome, our gene producing a specific protein and function in our body. Here's just a list of a few of the more common ALS-associated genes that have specific roles. So protein transport, RNA metabolism, mitochondrial function, all which are really important for typical healthy motor neuron functioning. Okay. So when we have a mutation in one of these genes, it causes that protein to dysfunction. The typical functionality of that gene isn't working anymore, and we have motor neuron degeneration. My clicker's not working. So this is just big picture of ALS gene discovery over time. So here on this axis here is our timeline, and here on this axis is our genes. So back in the 1990s, we had the first gene discovered and associated with ALS, which is SOD1. And these circles, the size of the circles represent the contribution to disease. So the larger the circle, the more common the gene is. So the 2 biggest circles on this graph here are SOD1, which is the second most common gene associated with disease. The largest circle here is C9orf72. We call it C9 for short, which is the most common genetic cause of ALS, which was discovered in 2011. This is a little bit old of a publication. I think it's a nice publication, but there's been some gene discovery since then, and a lot more learned about these specific genes. I'm going to focus a little bit at the start of my presentation on these more common ALS-associated genes, but you'll see here there's all these smaller genetic contributors to disease as well that are also really important that we understand how they function in the body. And at the end, I'll mention some of our drug development progress on those genes as well. So I'll just go through a couple examples. And so this is the point of this is to really give you context. Imagine going through genetic testing, having an abnormal result. What will that tell us? What it won't tell us? So just giving some better context into a few of the examples for the more common genes. So I mentioned C9orf72. C9 is the most common genetic cause of ALS. It's also the most common genetic cause for a certain form of dementia called frontotemporal dementia, FTD. So if you're getting genetic testing, one of the incidental findings on genetic testing could be that we identify a mutation in a gene that we think is causing ALS. it helps us understand why you have ALS. But it also tells us that you might be at risk for developing dementia in the future. So this would be true for the C, if you have a mutation in C9, TAR, DBP, and there's a number of other genes that can cause both ALS and dementia. Okay. So that's a consideration, is we're looking for a cause for ALS, we might find out something else too. It's when we test for the mutation within C9, remember I said it was like a spell checker? This genetic test is a little bit different than a spell checker. There's one specific mutation or abnormality at the start of the C9 gene called a repeat expansion. So the typical spelling of the C9 gene in this location has a 6-letter DNA sequence called GGGGCC. And typically that 6-letter sequence is just repeated a few times, maybe 2 times, maybe 5 times. Under 30 times is normal. That gene is able to be read properly and produce a proper protein if that 6-letter sequence is only repeated under 30 times. Okay. The mutation occurs when there's an expansion of that 6-letter sequence. So instead of it being repeated 2 times, it's 100 or 1,000 times. So you can think about it like this big clump at the beginning of the sentence, and we're unable to read past that because there's that big misspelling. And so with that, we have an abnormal toxic protein that's produced that causes motor neuron degeneration. Similar to many of the other associated genes, this is dominant, inherited in a dominant manner. And one of the things I'll talk about in a few slides is this terminology called penetrance. So when somebody has a mutation and has ALS, this mutation explains their disease. When we think about risks for family members, if your child, if your sibling gets tested and has this mutation, what's the risk for them? And so that refers to penetrance. What's the chance that somebody develops disease symptoms if they have an associated mutation? So we'll talk about that in a few slides. For C9, it's reduced, as with many of the other ALS-associated mutations. But I'm going to circle back to that in a minute. The other gene that I'll highlight is SOD1. This gene was first identified to be associated with ALS in the 90s, as I mentioned. It's the second most common genetic cause of ALS. It's less likely associated with dementia. So if I see somebody with a family history of ALS and frontotemporal dementia, I'm thinking more C9 or one of those other genes that are associated with both, rather than SOD1 is usually not strongly associated with FTD. This also has autosomal dominant inheritance, 50% chance that family members have the mutation if you yourself are identified with an SOD1 mutation. So SOD1 has over 100 different variants, disease-causing mutations that have been identified. So this is like the long sentence of the DNA sequence, and you can have a mutation at any different location within this long SOD1 sentence. Okay. So 2 examples that I'll talk about in regard to that penetrance is a mutation called A5V at position 5. We have a DNA nucleotide change. And another one further along in the gene called I114T, so at position 114. Okay, so just giving us some reference to when I start to talk about penetrance is even a mutation, 2 different mutations within the same gene can be cause very different disease risks. All right. So we've kind of gone over big picture genetics and a few examples of C9 and SOD1. One of the things I want to highlight here is the more recent publication that has stated evidence-based consensus, a consensus guidelines published from ALS neurologists and genetic counselors around the US. for genetic testing and genetic counseling for ALS. And the highlight takeaway of this long paper here is that all persons with ALS should be offered genetic testing for at least these genes, C9, SOD1, FUS, and TIR-DBP, the most common ALS genes. And then genes that also have that are strongly associated with ALS, and any gene that comes up with an FDA-approved therapy. Okay, so these are now published recommendations. Doctors, your neurologist should know about this, should be offering clinical genetic testing. And it's up to you, the person with ALS, if that's something you want to pursue. This is information you want to learn. So hopefully by the end of our conversation today, we'll have talked about some of those considerations for moving forward with testing. How is testing performed? Frankly, it's really simple these days. DNA can be identified in our, most all of our cell types. So we can get a saliva sample or a blood sample. If it's saliva, the lab will send a kit to your house. You'll spit into a tube, package it back up, and ship it back to the lab. So from there, we're able to isolate your DNA. We're able to run it through a number of different genetic tests. We'll analyze those genetic tests, and the laboratory will report out results. And from that, we can have a number of different results. A positive result, meaning we found a mutation. We believe that's the cause of your disease. We talk about what that means for you. Are there any clinical drug trials? Would this change your medical care at all? We talk about what that means for family members. A negative result means you don't have any genetic mutations in these ALS-associated genes. You don't have a known genetic form of ALS. This reduces the chance that family members are at an increased risk for disease. There's a third type of result called an uncertain result, which means we identified one of those spelling mistakes or changes when we read through the gene. But we don't have enough evidence to say, is that variation in your DNA code harmful to the gene function? Or is that just normal, benign variation that makes you a unique and different person from the next? Many of the uncertain results that we get back on ALS genetic testing, we can typically determine are likely benign and unrelated to disease. But it does just take a little bit more investigation on the genetic counselor, the neurologist part to figure that out. So I wouldn't be overly concerned with an uncertain result. These are pretty common. Your neurologist or genetic counselor will explain that in better detail to you. Okay. So what is your genetic risk? Usually when I meet with somebody for genetic counseling, one of the first things that I do is talk about their family history. And that helps me cater exact risk or what result could we get from genetic testing and cater that information specifically to them. So we start by understanding your family history. The majority of people with ALS have what we call sporadic disease. 90% of people have sporadic disease, whereas about 10% of people have what we call familial disease. Okay. So sporadic or singleton disease means that there's no family history of ALS. This makes up about 85 to 90% of people with ALS. And when we do genetic testing on people with no family history, it's usually negative, okay? Only about 5 to 10% of people with no family history have a positive result, okay? So family history is helpful. If you have no family history, I'd say there's a lower chance that we're gonna get an abnormal result. But it's not perfect. Some of these genes can hide in the family. And so because this is not zero, because we can still get an abnormal result 5 to 10% of the time, we still offer genetic testing to everybody regardless of a family history. So if testing is negative, if you don't have a genetic cause, What is the cause? Such a good question. It's a question I get from everybody who has negative results. It's one we don't have a good answer to right now. The specific cause is undetermined. What I say is it's likely a combination of small genetic factors that are in combination with multiple small environmental factors that in combination over time contribute to the onset of motor neuron disease symptoms. And this is something that's being investigated in a few different observational studies, but is really difficult to determine. And so we'll continue to take time to better pinpoint what those small genetic causes or environmental causes are. So this is different from familial ALS. So sporadic and familial are two terms that you might see historically in publications. Familial refers to having a family history, so 2 or more family members with ALS. And when we ask about a family history, we also ask about other related neurological diseases, so frontotemporal dementia or general dementia. Sometimes when we go back in time, somebody says that there was Alzheimer's disease in the family, but really perhaps that was misdiagnosed as frontotemporal dementia. I also ask about Parkinson's disease, which can co-occur with some of the genes that cause disease, and psychiatric diagnoses like schizophrenia. FTD is a much more behavioral dementia. So changes to personality, mood, irritability, there's a lack of inhibition. So those are some of the symptoms, and it can present, there can be memory loss, so it can present more like a memory-based dementia, and there can be psychiatric symptoms, so it can present sometimes like more of a schizophrenia. So those are the family history questions that we'll be asking to assess. Is there a family history of ALS or related neurological condition? If yes, 2 or more on the same side of the family, that looks like it's in a dominantly inherited pattern, we say, okay, there's an increased chance that this might be genetic in nature. So about 10 to 15% of people with ALS have this family history. When you do have this family history, we're typically able to identify a genetic cause. You're more likely to have a gene mutation if you have a family history of disease. It's about 75–80% of the time. Okay, so what's your risk? Here's back to our example of an individual here. You have ALS, you get tested, you have 2 normal gene copies, and so you either pass down a normal gene copy or another normal gene copy to your children, are not at an increased risk. So sporadic ALS means there's no family history that's been confirmed by normal genetic testing. Okay. One thing I'll get back to is, we're focusing on the genetics of ALS, the abnormalities that we might find on testing. And when we get into the considerations of genetic testing, I want us to think back on this negative normal result, because the majority of people don't have a family history. The majority of people do have negative results. And so sometimes this negative result can be really helpful too. In the context of understanding your disease, your management of disease and risks for family members. So if you have a positive result, again, it's that dominant inheritance, 50% chance it's passed down, a 50% chance it's not passed down, that your child inherits your normal gene copy. So the next slides I'll get into will be about that penetrance. So really talking about if a family member is found to have the same gene mutation, they're healthy, well, what's their risk for actually developing disease themselves? So to talk about penetrance and additional risk here, I like to bring out kind of this jar example. that helps in kind of this conversation. So in this jar, we have everybody has a baseline population risk for developing ALS. So we don't start at 0, we kind of start at this small baseline. And if we fill the jar to this line here, that is the point at which somebody develops disease symptoms. So we all might be exposed to either environmental factors or have genetic factors that could contribute to ALS. So many people might have these, but it might not be to the threshold in which they're actually developing disease symptoms. Okay. So for people with sporadic disease, no family history, and for those who develop ALS, like I said before, it's likely a combination of those small environmental factors plus small genetic factors that over time contribute to reaching to that threshold in which they are developing disease symptoms. This is different from the genetic or familial ALS. So I'll give some examples here. The first I'm going to talk about is that SOD1A5V variant, the one at the beginning of that SOD1 gene. This is known to be a more highly penetrant variant. So in our jar example here, people are born with a large genetic risk, okay? And so this puts them almost at this threshold of guaranteed ALS development in their lifetime. And so it's almost 100% penetrant, meaning almost everybody who has this mutation will develop disease symptoms in their lifetime. It's known to be earlier onset, can occur in the 30s or 40s, and more quickly progressive. Okay. So perhaps there's some people who escape that, who don't develop disease symptoms. So it's not 100% penetrant, but it is a highly penetrant mutation. This is different from that SOT1I114 variant, which has more reduced penetrance. So this genetic contribution, this gene mutation that they're born with, it puts them at an increased risk for developing ALS, above the baseline, above sporadic ALS, but lower than that compared to that larger, highly penetrant A5V variant. So perhaps some people will not develop ALS. Maybe they don't have those additional, genetic or environmental factors and don't reach the threshold of developing symptoms. But other people, indeed, this is still large enough to over time develop disease symptoms. So many of the genes associated with ALS have what we call variable or reduced penetrance. Penetrance meaning it's the proportion of individuals. If you have a mutation, what's the likelihood that somebody will express the symptoms associated with that mutation? So not everybody who has a genetic mutation may develop ALS in their lifetime. So for C9, chance of having the mutation are 50% if a family member is identified with the mutation. But the risk for developing disease is not 100%. So if C9 has reduced penetrance as well, it's not 100%. It's more similar to that SOD1A5V variant, SOD1 I114T, the third example on my last slide. Okay. All right. So, what I was showing here in this last slide is a sporadic family history. So sometimes, and this can be, those reduced penetrant variants can be really good examples of why some people with no family history still test positive for a mutation. So this is somebody with ALS. This is men represent squares, females represent circles. This is a person with ALS, their sister, their 2 children, mother, father, etc. So when we took this person's family history, nobody else, that black circle represents disease, nobody else had ALS. And so we thought this was presumably sporadic. But this individual also has the mutation, but doesn't have disease symptoms. So it's a mutation in a gene with reduced penetrance, and that's how these mutations, they say, can kind of hide in the family. Not everybody develops disease symptoms themselves. Okay, moving into considerations for genetic testing, there's a number of these. So we'll kind of go on either side what people might consider in making the choice to proceed with testing. So testing can help determine an underlying cause if we have a positive result. If negative, it might not help us any further. We might be where we were before we got genetic testing. It can help refine the prognosis. So for some specific mutations, we know that they're more slowly progressive or more quickly progressive. So if we know that about the specific mutation that's been resulted, it might give us more insight about what the future of disease might look like. If we get a negative result, of course, that might not be the case. Or if we get a result in a gene we don't know a lot about, that might not help us in that regard as well. Testing can help clarify our risks to family members. So if we get a positive result, it could, refine that family members indeed are at increased risk. It's dominant inheritance, but it's in a reduced penetrant gene. Okay. And this is when I'm circling back to that negative result. So focusing on the fact that most people don't have a family history, most people are sporadic, most people have a negative result. And so I think a lot of our discussion focuses on this positive abnormal result, but there's a lot of people that will reach out to me now who say, my parent had ALS 10 years ago and didn't get genetic testing. They didn't have a family history, but I just want to know for sure, was their disease genetic or not? So speaking to your kids, speaking to your family members about, will it be helpful too to get a negative result? Can that be reassuring for them as well, that they're not at an increased risk? I think it is an important way to kind of flip this on the head and think about it as well. Of course, that has to be balanced with the anxieties that can come along with, the smaller chance that we do find an abnormal genetic result. So balancing those two is kind of important and individualized to you and your family. The main reason why, one of the main reasons why these guidelines came about and why most physicians are and should be offering genetic testing is the newly FDA-approved treatment for SOD1 called Qalsody. So anyone with an SOD1 mutation, will discuss the option for treatment with Qalsody. And I think I get into that in another slide. So if you have a mutation in this gene, it could change how we care for you. On the flip side, the results, if in a different gene or if negative, really might not change your care plan. When, we have a number of different clinical trials that are genetic specific or non-genetic specific or sporadic. And so having genetic testing kind of opens the door to either talk about or not talk about those genetic clinical trials. So the results of testing can help inform options for different clinical trial enrollment. And the flip side of that is that, of course, clinical trials, whether it's genetic or not, all carry a risk. So again, these are considerations if you're considering enrollment in a clinical trial, something to think about here. I will jump from here to talk about some of these clinical trials and gene-targeted therapies. So that's kind of what I'll focus on in my next part of the talk here. So one of the main types of therapeutics that are more common and going through clinical trial for some of these ALS genes are antisense oligonucleotide therapies, or ASO therapies. So just a brief overview of ASO therapies. Again, this is our DNA strand that's translated to messenger RNA, which creates a typical protein or function in our body. So that's what we reviewed in those first slides. When we have a mutation in our DNA, it creates mutated RNA and a disease-causing protein. ASO drugs are designed to target and latch on to your RNA strand. In many of the ALS-associated genes, they're toxic, and they're gain-of-function mutations. They create an abnormal toxic protein. And so this ASO drug is basically, I like to think about it as a tag. It latches on to your mRNA and kind of puts a stop signal. So it cannot create that toxic protein that typically leads to motor neuron disease. So we're putting a stop to that cause of motor neuron dysfunction. Okay. So that's big picture simplified ASO therapy. Okay. Qalsody is an ASO drug to target and treat the SOD1 gene. Okay, so people with SOD1 mutations, this was approved, I think, spring of 2023. Yes, spring of 2023. So it's been approved for a little over a year now. I think we have a number of people at Columbia who are receiving treatment for Qalsody. The other exciting piece of this is the company who developed Qalsody has now launched a pre-symptomatic trial for treating people who have this mutation but don't have disease symptoms. So perhaps the children or siblings of people with SOD1 ALS are enrolling in this ATLAS trial. They're being monitored over time, and kind of at a threshold point in which they might develop disease symptoms, they're going on a treatment arm of the clinical trial to receive the drug pre-symptomatically with the aims to prevent disease onset and progression. So this is very exciting for the SOD1 community. And I always like to take us back into the big picture of things. This is exciting for SOD1, and it's not happening for the other ALS-associated genes yet, right? But if we think about what's to come in 5 years, in 10 years, in 20 years, as your children get older, I want us to think about the families who got tested for SOD1 10 years ago, and now their children are benefiting from this now. So important to put into context is what can be helpful now, what is true now, and what might be true, what opportunities might be available down the line. Clinical trials, kind of an overview. I mentioned Qalsody was approved last year. The ATLAS study is ongoing. There's an ASO designed for another common ALS mutation called FUS, so the fusion trial that's in actively recruiting. There was an ataxin-2 ASO. I believe that trial was recently terminated. And then there also was a C9 ASO trial, which failed to show clinical benefit and was also discontinued. So I know that there's more preclinical work, meaning that more laboratory and drug development studies are occurring now that hopefully will go into clinical trial for these more common genes in the next few years. Additionally, we at Columbia have launched a program called Silence ALS, and this is a program coupled with the NLORM Foundation, excuse me, and NLORM Foundation is a nonprofit foundation who aims to develop ASO drugs for rare genetic diseases. And we've partnered with them to focus on the development of ASO drugs for rare ALS, rare genetic forms of ALS. So we've already developed an ASO for the CHCHC10 gene, same with a few mutations in TAR-DBP. We're working on more of that, as well as mutations in annexin 11, PFN1, KIF5A, VCP, VAPB. So the list of potential opportunities are beginning to grow as these types of therapies become more, better learned, better established for other genetic diseases. So again, I want us to think about what's available now and what might be available for you in the future, for your family in the future, and how can this genetic information be beneficial to you or your family, both a positive result if we have a mutation here, or a negative result. Okay. So considerations for testing, a few more that I want to touch on that we talk about in our genetic counseling sessions. The first is informing yourself or family members for family planning options. And then we always talk about potential insurance discrimination risks. Okay. So family planning consideration, here's again a pedigree of family history. An individual with ALS is known to have a positive gene mutation. Their daughter in this case gets tested, also is asymptomatic but known to have the mutation. There's now genetic technology to and ensure that a future pregnancy doesn't have that same gene mutation, that this individual doesn't pass it down to the next generation. So this is the process of using in vitro fertilization. So a woman is stimulated with hormone therapies to overproduce the number of eggs in a cycle. Eggs are retrieved. They are fertilized with sperm to create an embryo. That embryo is grown in culture until it reaches a certain number of cells. And then it can be transferred back into a woman, into the uterus to produce a pregnancy, and a fetus is grown. Okay, so that's typical in vitro fertilization process. PGTM stands for pre-implantation genetic testing for a mutation. So at this stage, the embryo is biopsied. We take a little sample of the embryo, DNA sample of the embryo, and we do genetic testing on this embryo, on all of the embryos that were created. And then we only implant an embryo that doesn't carry that known mutation. So this person is ensuring that that mutation isn't passed down to the next generation. So especially, this can be true for younger individuals with ALS who are currently family planning, or for children who are actively family planning and want to make sure, would want to make family planning decisions based on genetic information. So that's an option in terms of considerations here. Okay, last piece here is that insurance discrimination piece. So there's a law in place called GINA, Genetic Information Non-Discrimination Act. And this law protects all of us against discrimination from our health insurance companies and our employers if we work at a company more than 15 people. So for example, if I have a cancer gene mutation, I'm at risk for developing cancer in the future. My health insurance still has to cover all my healthcare cancer-related needs. My employer, Columbia, can't fire me because they know I might develop cancer in the future. Okay. The limitations of GINA are that it does not protect against life insurance, long-term care insurance, or disability insurance. Okay. So for people I'm talking to who have a diagnosis of ALS that's documented in this medical records, I kind of say that this is a bit of a moot point. If you apply for life insurance right now, you'll likely be discriminated against based on your diagnosis of ALS. And the addition of a genetic result to this isn't going to, doesn't really matter in terms of life insurance genetic discrimination risk. Okay. The big thing where we really talk about GINA and insurance risks are if you test positive and your child wants to get tested themselves, they would be at risk. Okay. And so before your child gets genetic testing, we make sure they're properly insured, have life, long-term care, disability in place, or however they want that for their future, before they get tested themselves. So most of the time I say, we cross this bridge once we come to it. If you test positive, if your daughter wants to get tested, before she gets tested, we'll make sure those things are in place. Okay. So, oops, that's what that bullet point says. Again, kind of a moot point for people who are going through diagnostic testing if you have ALS, and more of a conversation that we talk about, if asymptomatic family members are getting tested. Okay. So here's kind of that summary of of considerations for testing. A few things I've taken off here are costs of genetic testing that can be prohibitive. There's a number of different sponsored testing programs that offer free genetic testing right now. So I've taken that off of the list because I really don't think it should be a barrier or a big consideration in testing anymore. And I've kind of italicized that insurance discrimination risk because, again, for someone who's getting diagnostic testing, it's kind of a moot point. And, you know, we deal with that with family members. We do that before testing occurs. Okay. I believe that's the conclusion of my slide. So it looks like we have some time left, and I think we have an hour here, so we have some time left, and I'm happy to answer any questions from the group or open it up for discussion.
All right, Ellie, thank you so much. That was really informative and a lot of also food for thought. If anyone has any questions, please add them to the Q&A box. One question is if and the attendees today are not just in the New York City area, it's ALS United, so spread far and wide. If they do get tested, how can they also have a genetic counselor, find a genetic counselor?
Good question. So National Society of Genetic Counselors, nsgc.org. There's a patient list, or there's kind of a portal in which you can say, find a genetic counselor. And so you'll say where you live, you want to find a neurogenetic counselor, and that will loop you into a more local neurogenetic counselor. That's one way to go about it. Also, for some of the tests that are ordered at Prevention Genetics and at Invitae, two of the more common, bigger testing companies, your doctor can click referral to a genetic counselor, and there's services for which they can provide genetic counseling alongside doing the genetic testing.
Okay, great. What is the current penetrance rate or percentage for C9, the risk of disease if you have the C9 gene?
Yeah, it's a really good question. The quick answer is there's no percentage right now that is published that we can state the penetrance is X%. Okay. I'll tell you a little bit about the history though, and hopefully that will provide some context as to where we are, what we know, and what we don't know. When the mutation was first discovered and original families were identified with the mutation, publications between 2012 and 2017 say that its penetrance of C9 is high, almost complete, or 90% penetrant. Okay. Those publications, a lot of them were looking at, highly penetrant families, large families where a lot of people are developing ALS. And I believe some of them kind of lacked, the numbers of that we have today in some of our studies. There's more recent publications in 2023 and 2024 that look at how common the C9 mutation is in a healthy population. How common is the mutation around the world? So the incidence of C9, of people having the C9 mutation, compared to how many people are actually developing C9 ALS. So more of a comparative analysis, which is a different penetrance calculation. And those calculations were much lower, like 30 or 40% penetrant. Okay. Those publications, the caveat here is that they didn't include a family history or personal history of dementia. Okay. So I believe that those are kind of on the low side. 30% might be low on penetrance because those are biased. They don't include dementia in their penetrance calculation. And, the 90% is on the high side. I think, they weren't pulling from enough data for those to be accurate. So it's definitely not 100%. We know that it has reduced penetrance. more data will be published, I think, in the coming years around this, as more of those studies with, larger population genetics. But I would say probably anywhere between like 50 and 80% penetrant. And hopefully, as time goes on, there's clarification of that penetrance number. Maybe it's clarified for ALS and FTD. And it really might also involve what your family history looks like. So a catered penetrance number based on what penetrance looks like within your own family. And so those are things that are still need to be better refined. And I don't want us to take exact penetrance numbers from my talk today because it's still being investigated.
Okay, all right, thanks, Ellie. All right, we have another question. Can people with familial receive genetic testing outside of insurance so it is not reported?
Yeah, so there's kind of two pieces to this, which isn't clear from the question. The first is if you have ALS and you're getting genetic testing, is this in the chat? No, it's not. So
in the Q&A, but it's
okay.
So
Oh,
familial members are asymptomatic.
Familial members, asymptomatic family members, yes. So there's a study ongoing that's being launched now at 34 sites around the US called Prevent ALS. This study is an observational study of asymptomatic individuals at risk for developing disease, at risk for having a mutation. And through the Prevent study, we offer genetic counseling and genetic testing outside of the insurance network system. And in a de-identified manner, your name's not on the report. So that's kind of the best way I would say to access pre-symptomatic or asymptomatic genetic testing without insurance. Columbia is a site for that. There's many sites around the US. Myself and 2 other genetic counselors perform the genetic counseling and testing for all the different sites. So you can reach out to me or you can, and I'm happy to connect you with your local site to talk about that further.
So Ellie, just for clarification, that it is still possible to do anonymous testing if I was to pay for the testing myself and do like a saliva test at home. Is that possible?
That depends on the genetic counselor and on like the hospital practices. So if you say, I don't want to be involved in research, but I still want to get anonymous genetic testing and I want to pay for it myself, it's probably possible. The exact protocol will depend on the hospital, the doctor, and the genetic counselor.
Okay, all right.
But you'll likely have to pay out of pocket for the cost of the test and out of pocket for the doctor and genetic counselor visit.
So expenses can add up doing it that way.
I'd still say it's under $1,000 in total, but it will be of cost likely.
Okay, thank you. All right, in the Q&A box as well, there's another question. My dad has an extremely progressive form of ALS and is at the end stage. Symptoms started in August. Is there any connection to progression and having the familial type of ALS? Also, thank you for talking about the insurance. I plan to get long-term care insurance and will do so before testing. Lastly, can someone send information to testing places that we can possibly use?
Yeah. So first, I would say if you are thinking about getting testing yourself, it's important that your dad first receive genetic testing, right? Unless you have a strong family history, multiple other family members beside dad with ALS, you yourself as a healthy individual with one family member with ALS are not recommended to have testing. So first priority is getting dad tested. He can speak with his neurologist who should be offered genetic testing. There are sponsored programs through Prevention Genetics and Invitae that your doctor can order from that are free of charge, okay? Or his doctor can order from. They can do an at-home saliva kit to your dad so that they don't have to come back in to the medical center, okay? There are some genetic forms that are more associated with quickly progressive disease, but I would say, ALS can also be a quickly progressive disease. There's a lot of variability within the disease spectrum. So just because your dad has a quickly progressive form of ALS doesn't make me concerned that this is genetic. What would make me concerned is if there's multiple other family members also with ALS. So I would say if you're thinking about genetic testing yourself, the first step is definitely getting dad testing. If dad's testing is negative, testing for you is not warranted. It would not be done. If dad's testing is abnormal, then, think about life insurance, long-term care, and then you can get tested yourself. Did I answer everything? I know there was a couple questions in there.
Yeah, I think the last one is just sending information to testing places that can be used by the attendees here.
Yeah, I would just say Google prevention genetics, ALS sponsored testing. Here, I'll put it in here. Am I allowed to type? Oh, I'm not allowed to type. Prevention genetics, ALS sponsored testing, or INVITAE, INVITAE, ALS sponsored testing. So sponsored testing is the keyword. That's the free genetic testing. Those will hopefully give you those direct links to the options there. But again, the neurologist should be able to order those tests, diagnostic ALS testing.
Okay. One final question, I think, before to end our time here today. If someone is interested, but a little scared to get tested, can they meet with a genetic counselor ahead of time to discuss pros and cons and help make their decision?
Yeah, absolutely. So hopefully, hopefully there's a genetic counselor on staff at the site in which you're being seen. Sometimes there might not be a genetic counselor directly working in your clinic, but there should be genetic counselors at, the larger medical center. So if you're really wanting to speak to a genetic counselor about pros and cons, kind of push your doctor to say, are there any genetic counselors at the hospital that can speak with me about, genetic testing? And then also, when the doctor places the order for the prevention, I know it's true for prevention, that prevention sponsored panel, they can click a box that says patient wants to speak with genetic counselor. And so prevention genetics would connect you with a genetic counselor. So yes, there are a few different ways to go about it. I'd first start to talk with your neurologist, see if there's any resources at your local institution. And then the other route would be through the prevention genetic counseling offerings that they have.
Okay, wonderful. Thank you so much for your time today and your presentation. It was very helpful. I'm glad we recorded it so we could watch it back again. Do you have anything final thoughts before we close out?
No, I think, there's a lot of considerations to go into testing. It's definitely a recommendation now for diagnostic genetic testing again, but that information, needs to be catered to you and your family and your individual decisions. So I think it's important that people are informed about benefits and drawbacks and take that information and cater it to what they see for themselves right now, opportunities, clinical trials right now, but also important to think about what might be true for the future as well, because the genetic field is indeed changing, and so I think that's an important piece that's often missed in kind of the emergent thinking around genetic testing.
Excellent. Well, thank you so much for today's presentation. Thank you for everyone for attending, and we look forward to seeing you at our next Talk, Learn, and Connect seminar that we provide at ALS United Greater New York. Thanks, Allie. Thank you very much.
Thanks so much. Have a nice day, everyone.
Take care. Bye.